Hello there, welcome once more to this YouTube channel. Kindly subscribe to this channel. The topic today is embryology of central nervous system. Embryology of central nervous system. This is part 1 of this lecture. The central nervous system appears at the beginning of the 3rd week as a slipper-shaped plate of thickened ectoderm. This is what you call the neural plate. And in the middorsal, you find that in the middorsal region in front of the primitive node. Its lateral edges soon start to elevate to form what we call the neural folds. With further development, that as development goes on, the neural folds continue to elevate. The neural folds approach each other in the midline, and finally what you see is fusion of the neural folds, and when they fuse they form the neural tube. Right here is an illustration of neural plate formation, neural tube, and how the neural folds, and finally you see neural tube formation. You can see also the neural crest cells there, the neural crest cells, and the neural crest cells you find them between the neural tube that is formed and the epidermis, and the neural crest cells they migrate laterally as the neural tube is formed. Fusion of the neural tube begins in the cervical region and proceeds in cephalic and caudal directions. Once fusion is initiated, the open ends of the neural tube forms the cranial and caudal neuropores, and this communicates with the overlying amniotic cavity. You must not forget that the closure of the cranial neuropore proceeds cranially from the initial closure site in the cervical region, and then from a site in the forebrain that forms later. This later site proceeds cranially to close the most rostral region of the neural tube, and it goes caudally to meet the advancing closure from the cervical site. Final closure of the cranial neuropore occurs at the 18 to 20 summit stage. That's at the 25th day of development, intrauterine development. Closure of the caudal neuropore occurs approximately 2 days after the closure of the cranial neuropore. Don't forget also that the cephalic end of the neural tube shows 3 dilatations. You must not forget that. There are 3 dilatations of the cephalic end of the neural tube. These 3 dilatations are the primary brain vesicles, and the primary brain vesicles are namely 1, the prosencephalon or the forebrain. The second one is the mesencephalon or the midbrain, and of course the third one, the last but not the least, is the rhombencephalon or the hindbrain. At the same time, you also notice 2 flexures. You see 2 flexures. You have a cervical flexure at the junction of the hindbrain and the spinal cord. You also have a cephalic flexure at around the midbrain region. Now, when the embryo is 5 weeks old, the prosencephalon consists of 2 parts. You see a part we call the telecephalon, which is formed by a mid portion and 2 lateral outpocketings, what we call the primitive cerebral hemispheres. So the second part of the prosencephalon is the diencephalon, which is seen as outgrowth of it presents outgrowth of the optic vesicles. You have a deep furrow, the rhombencephalic isthmus, which separates the mesencephalon from the rhombencephalon. The rhombencephalon also consists of two parts, what we call the mesencephalon, which later forms the pons and cerebellum. The second part of the rhombencephalon is the mesencephalon. The boundary between these two portions is marked by what we call the pontine flexure. Flexure is like a bend. Then the lumen of the spinal cord, that's the central canal, is continuous with that of the brain vesicles. The cavity of the rhombencephalon forms the fourth ventricle. That of the diencephalon is the third ventricle, and those of the cerebral hemispheres are the lateral ventricles. The lumen of the mesencephalon connects the third and fourth ventricles. Then you must know that this lumen becomes very narrow and is then called the aqueduct of Sylvius, what some call the cerebral aqueduct. The lateral ventricles communicate with the third ventricle through what we call the interventricular foramina of Monro. Right here is an illustration of the primary brain vesicles. You can see the prosencephalon, the mesencephalon, and the rhombencephalon. Right there also you can see the 5 secondary brain vesicles. That's the prosencephalon differentiating there, and it's differentiated to give you the telencephalon and the diencephalon. The adult brain structures that come from the telencephalon is illustrated right there in the diagram. You can see the cerebral hemispheres, that's the cortex, the white matter, and the basal nuclei. You find all that in the adults. These are structures that come from the telencephalon. Then the neural canal regions of the telencephalon finally differentiates to give you the lateral ventricles, and the superior portion of the third ventricles. Still looking at the diagram, you can see the diencephalon there with the optic vesicles as outgrowths there. The diencephalon differentiates to give you the thalamus, the hypothalamus, and epithalamus. Take a look at this diagram. You see that the adult canal region of neural canal regions, that region you find in that differentiates in the diencephalon, is the third ventricle. Most of the third ventricle actually forms there. Now we're still looking at the diagram. Let's see the mesencephalon. The mesencephalon is differentiating there to give you the adult brain structures. That's the brain stem, part of the brain stem that we call the midbrain. The adult neural canal region you find in the midbrain is the cerebral aqueduct. I said that earlier on. I'm still looking at the diagram. You see the mesencephalon, which is a part of the rhombencephalon. The rhombencephalon differentiates to give you the mesencephalon and the myelencephalon. The mesencephalon can be seen there, differentiating to give you finally adult brain structures like the pons, which is a part of the brain stem, and then the cerebellum. And the adult neural canal region you find there is the fourth ventricle. Also, the myelencephalon, which is the second part that differentiates from the rhombencephalon, is right there in this diagram. It differentiates to give you a part of the brain stem, that's the medulla oblongata. Then you have the adult neural canal region there is also the fourth ventricle. So that is a diagram right there illustrating the differentiation of the brain from the primary brain vesicles to the 3 primary brain vesicles to 5 secondary brain vesicles that finally differentiates to give you the different part of the brain. Now right here is an illustration of the development of the spinal cord. You can see the marginal layer which is the outmost layer. The mantle layer is deep to that, and the deepest is the ependymal layer. Then the neural canal is right there in the center. This is it's a cross section of the neural tube. That is what you're seeing here. It will eventually differentiate to give you an alar plate anteriorly, and then basal plate at the alar plate is actually posterior, and the basal plate anterior. These are structures that differentiate to form the spinal cord. And of course, between the alar plate, which is posterior, and the basal plate, which is ventral or anterior, is the sulcus limitans. It's right there illustrated in the diagram. Then finally, these structures differentiate to give you the gray matter. The alar and basal plates will differentiate to give you the gray matter. You can see the alar and basal plate are colored yellow. They differentiate from the mantle layer, which is colored yellow there in the first diagram. And you have the mantle layer differentiating to give you the alar plate and the basal plate, which eventually will form the gray matter of the spinal cord. The alar plate will differentiate to give you the dorsal horn, while the basal plate differentiates to give you the ventral horn. And posteriorly, you have a dorsal median groove, which forms there between the right and left parts. That forms through the marginal layer. And also anteriorly, you have a ventral fissure forming through the marginal layer also. So I'm talking about the development of the embryology of the spinal cord. There are some structures you must not forget. We talk about the neuroepithelial, mantle and marginal layers, then the epidermal layers. These are all layers you must not forget. So as far as embryology of the spinal cord is concerned, remember that the wall of a recently closed neural tube consists of neuroepithelial cells. These cells extend over the entire thickness of the wall and form a thick pseudostratified epithelium. You have junctional complexes at the lumen which connect the cells. During the neural groove stage, during the formation of the neural tube, you have the neural plates. As it folds, as you have the neural folds by the side, you have a neural groove formed there. So at the neural groove stage, and immediately after closure of the neural tube, you have a division of the cells, the neuroepithelial cells. They divide rapidly and they produce more and more neuroepithelial cells. Collectively, they constitute the neuroepithelial layer or the neuroepithelium. Once the neural tube closes, the neuroepithelial cells begin to give rise to other cell types characterized by a large, round nucleus with pale nucleoplasm and a dark staining nucleolus. These are the primitive nerve cells or neuroblasts. So the neuroepithelial cells differentiate to give you the neuroblasts. They form the mantle layer. So you see these neuroblasts that are differentiating, they are arranged in the mantle layer, which is a zone around the neuroepithelial layer. The mantle layer later forms the gray matter of the spinal cord. I said that a while ago. The outmost layer of the spinal cord is actually the marginal layer, and it contains nerve fibers that differentiate and emerge from neuroblasts in the mantle layer. So as the neuroblasts are differentiating in the mantle layer, They also differentiate to give you nerve fibers that are arranged around the marginal layer. So the marginal layer is formed at the periphery of the mantle layer. Then you have myelination of the nerve fibers that you find in the marginal layer. And then when myelination occurs, you now have a whitish appearance, and you now call that the white matter of the spinal cord. So from the marginal layer, you're going to have the white matter of the spinal cord. Right here is an illustration of the development of the spinal cord. You can see the marginal layer, you can see the mantle layer, and of course the epidermal layer. I'm going to talk about differentiation of all these parts. I've already talked about how the marginal layer and the mantle layer differentiates. Then I mentioned the alar plate and the basal plate. I mentioned earlier on. They are differentiating from the mantle layer. So they're going to form the gray matter of the spinal cord. Then also here you have a roof plate and the floor plate. The roof plate and the floor plate. The floor plate is ventral while the roof plate is dorsal. So eventually at the midline of the ventral portion you have the floor plate, and eventually there you have a structure differentiates, which is then you have the roof plate. The roof plate is dorsal. It's found at the midline of the dorsal portion. So eventually you have the dorsal median groove differentiating there. So let's talk about the basal, the alar, the roof and floor plates. Right here you have an illustration of development of the spinal cord. You can see the lumen, the epidermal layer and the mantle layer. Eventually you see the differentiation to form the floor plate, roof plate I talked about, then the alar plate and the basal plate, and then sulcus limitans between that demarcates the ventral portion of the spinal cord from the dorsal portion. And eventually you have the spinal cord there, well differentiated as the C, and you have the gray matter. The gray matter has two colors there, and the anterior portion has a different color from the posterior portion. And then you can see the ventral fissure and the dorsal median groove. So you must, regarding the basal, the alar, the roof and floor plates, you must remember that as a result of continuous addition of neuroblasts to the mantle layer, each side of the neural tube shows a ventral and a dorsal thickening. The ventral thickening is what we call the basal plates you have by the right and the left, with which contains, this basal plate contains ventral horn cells. These ventral horn cells are motor cells, so we say they are ventral motor horn cells, and they form the motor areas of the spinal cord. The dorsal thickening is what we call the alar plates by the right and left. They form the sensory areas. You have a longitudinal groove, what we call the sulcus limitans, that marks the boundary between the alar plate and the dorsal and the basal plate. The dorsal and ventral midline portions of the neural tube are known as the roof and floor plates. Roof and floor plates. So you have at the dorsal portion you have the roof plate. The dorsal midline portion you have the roof plate. At the ventral midline portion you have the floor plate. They do not contain neuroblasts. They serve primarily as pathways for nerve fibers crossing from one side to the other. In addition to the ventral motor horn and the dorsal sensory horn, you have a group of neurons which accumulate between the two areas, so between the ventral and dorsal sensory horns. You have a group of neurons that form what we call the small intermediate horn. This intermediate horn contains neurons of the sympathetic portion of the autonomic nervous system. You find it present only at the thoracic region, T1 to T12. That's the region of the 1st to 12th thoracic vertebrae, and also at the upper lumbar levels. That's the level of the 2nd and 3rd lumbar vertebrae. So at those levels of the spinal cord, that is where you find the intermediate neurons. Right here is an illustration showing the roof plates and the floor plates, and then you can see the differentiation of the spinal cord and even part of the meninges surrounding the spinal cord. Note that as development is going on of the spinal cord, you have histological differentiation of the neural tissues. You have nerve cells. How do nerve cells come about? Neuroblasts or primitive nerve cells arise by division of the neuroepithelial cells. Initially, you find that they have a central process. The differentiating nerve cells have a central process extending to the lumen. So that one is like a transient dendrite. But when this developing nerve cell migrates into the mantle layer, you have this central process disappears, and the neuroblasts are now attains a structure such that it is round and apolar. With further differentiation, you now have the nerve cell with two cytoplasmic processes that appear on opposite sides of the cell body, and this form a bipolar neuroblast. The process at one end of the cell elongates rapidly to form the primitive axon, and the process at the other end shows a number of cytoplasmic processes, the primitive dendrite. The cell at this stage is known as a multipolar neuroblast. So you see the neuroblast, the nerve cell is differentiating. So the multipolar neuroblast goes on to differentiate further, and it becomes the adult nerve cell or neuron. So you must not forget, as once the neuroblast form, they lose the ability to divide. So you find out that the axon of neurons in the basal plate break through the marginal part, the marginal layer, and they become visible on the ventral aspect of the spinal cord. All these neurons form what we call the ventral motor root of the spinal nerve. So these axons that are coming out form the ventral motor root of the spinal nerve, and they are important because they conduct motor impulses from the spinal cord to the muscles. Don't forget, remember that you need to recall that a neuron is a structural and functional unit consisting of the cell body and all its processes. Now there is need to talk about the glial cells. The glial cells, the majority of primitive supporting cells, what we call the glioblasts, are formed by the neuroepithelial cells. After production of neuroblast, ceases. So as the neuroepithelial cells differentiate to form neuroblasts, once they are done with that, they differentiate also to form some other cells like the glioblasts that will differentiate to give you the glial cells. The glioblasts migrate from the neuroepithelial layer to the mantle layer and marginal layer. In the mantle layer, the glioblasts differentiate into protoplasmic astrocytes and fibrillar astrocytes. You also have another type of supporting cell which is most likely derived from the glioblast. That is called the oligodendroglial cell. The oligodendroglial cell. This one is important because this cell is found around the marginal layer. It forms myelin sheets around the ascending and descending axons in the marginal layer. So in the second half of development, you also see a third supporting cell, what we call the microglial cell. It appears in the central nervous system. This microglial cell is highly phagocytic and it's derived from mesenchyme. Then don't forget that when the neuroepithelial cells cease to produce neuroblast and glioblast, they differentiate into epidermal cells. The epidermal cells line the central canal of the spinal cord. Right here is an illustration of the neural crest cells. I need to talk about neural crest cells now. So we saw a diagram earlier on showing how the neural crest cells differentiate. So right here we want to see the neural crest cells differentiating. You see the diagram, the neural tube formation, neural plate formation before the neural tube formation. So and the neural crest cells, as the neural tube is formed, neural crest cells that are formed between the neural tube and the epidermis there, they move laterally and they differentiate to give you some other cells that we're going to talk about now. So during elevation of the neural plate, a group of cells appear along each edge. That's the crest of the neural folds. These cells are called the neural crest cells. They are ectodermal. Remember that neuroblasts, fibrillar and protoplasmic astrocytes and the epidermal mast cells originate from neuroepithelial cells. Microglial cells develop from mesenchyme cells. Now, the origin of the oligodendrogless cell is not clear. Some people believe it comes from the glioblast. The neural crest cells now that are formed, I'm talking about neural crest cells. Actually, the neural crest cells that you find differentiating between the neural tube that is formed and the epidermis migrate laterally and give rise to the sensory ganglia. That's the dorsal root ganglia of the spinal nerves and other cell types. So as development goes on, neuroblasts of the sensory ganglia form two processes. You have a centrally growing process which penetrates the dorsal portion of the neural tube. In the spinal cord, they either end in the dorsal horn or ascend through the marginal layer to one of the higher brain centers. So these processes now, we're talking of the processes that come from the neuroblast of the sensory ganglia. The processes that come from the neuroblast of the sensory ganglia. So these processes are known collectively as the dorsal sensory roots of the spinal nerve. The peripherally growing processes join fibers of the ventral motor roots and hence participate in formation of the trunk of spinal nerve. Eventually, these processes terminate in the sensory receptor organs that you find in the body. So, in other words, neuroblasts of the sensory ganglia are derived from neural crest cells and they give rise to the dorsal root neurons. In addition to forming sensory ganglia, cells of the neural crest also differentiate into sympathetic neuroblasts. So the neural crest also differentiate into sympathetic neuroblasts, the Schwann cells, pigment cells, odontoblasts, meninges, and mesenchyme of the pharyngeal arches. Right here is an illustration of a diagram to show how the neural crest cells differentiate the structures that are formed that I named a moment ago. Now let's see about the embryology of the spinal nerves. Don't forget that motor nerve fibers begin to appear in the 4th week, arising from nerve cells in the basal plates, that's the ventral horns of the spinal cord. These fibers collect into bundles, what are known as the ventral nerve roots. You also have the dorsal nerve roots, which forms as collection of fibers originating from cells in the dorsal root ganglia, what we call the spinal ganglia. Central processes from these ganglia, that's the dorsal root ganglia or spinal ganglia, form bundles that grow into the spinal cord opposite the dorsal horns. You also have distal processes which join the ventral nerve roots to form a spinal nerve. As soon as this occurs, as soon as the spinal nerve is formed, the spinal nerve divides into dorsal and ventral primary rami. The dorsal primary rami innervates the dorsal axial muscles, the vertebral joints, and the skin of the back. The ventral primary rami innervate the limbs and ventral body wall and form the major nerve plexuses, that's the brachial plexus and the lumbosacral plexus. Now I need to talk about myelination. You have Schwann cells. Remember, Schwann cells differentiate from the neural crest cells. So the Schwann cells myelinate the peripheral nerves. But these cells originate from neural crest cells, like we said earlier on. They migrate peripherally and wrap themselves around axons, forming neurolemma sheath. But at the 4th month of fetal life, many nerve fibers take up a whitish appearance. Why do they appear whitish? It's as a result of deposition of myelin, which is formed by repetitive coiling of the Schwann cell membrane around the axon. The myelin sheath surrounding nerve fibers in the spinal cord has a different origin, however, not from Schwann cells but from the oligodendroglial cells that I mentioned earlier on. So you have myelination of nerve fibers in the spinal cord beginning at the 4th month of intrauterine life, but some of the motor fibers descending from the higher brain center, so the spinal cord, do not become myelinated until the 1st year of postnatal life. It's interesting to even note that tracts in the nervous system, they become myelinated at about the time they start to function. Now let's talk about positional changes of the spinal cord. In the 3rd month of development, the spinal cord extends the entire length of the embryo, and spinal nerves pass through what we call the intervertebral foramina at their level of origin. But with increasing age, what happens? The vertebral column and the dura lengthen rapidly. They grow rapidly, more rapidly than the developing spinal cord. So they grow rapidly than compared to what the neural tube is doing, and then you have the terminal end of the spinal cord gradually shifting to a higher level. So at birth, the spinal cord ends at the level of the 3rd lumbar vertebra. As a result of this, you have this disproportionate growth between the vertebral column and the spinal cord. You have spinal nerves now running obliquely from their segment of origin in the spinal cord to get to the corresponding level of the vertebral column. The dura remains attached to the vertebral column at the coccygeal level. So what do you see in the adults? In the adult, the spinal cord terminates at the level of L2 to L3. But in the adult, the dural sac and subarachnoid space extends to the level of S2. That's the 2nd sacral segment. Below L2 to L3, that's the 2nd lumbar to 3rd lumbar segment, you have a thread-like extension of the pia mater, which forms the phylum terminale. The phylum terminale is attached to the periosteum of the 1st coccygeal vertebra, and it marks the tract of regression of the spinal cord. When you look at the phylum terminale, it reminds you and that this is actually about the regression of the spinal cord. Nerve fibers below the terminal end of the spinal cord constitute the what we call the caudal equina, and the cerebrospinal fluid is tapped during a lumbar puncture. When you need to tap cerebrospinal fluid during lumbar puncture, a needle is inserted at the lower lumbar level, and you try to, in doing this, you avoid the lower end of the spinal cord. If you're actually doing a lumbar puncture for a child, you have to go lower to the level of L4 to L5, so that you don't you avoid the spinal cord, you don't injure it. So you have to bear that the positional changes of the spinal cord in mind when you are doing lumbar puncture. Now, molecular regulation of the spinal cord development. The development of the spinal cord is regulated by certain genes and proteins factors. At the neural plate stage in the spinal cord region, the entire plate expresses transcription factors. You have some transcription factors, what we call the PAX3, PAX7, MSX1, and MSX2. All these contain a homeodomain. This expression pattern is altered by the sonic hedgehog protein that comes from which is expressed in the notochord, and also the bone morphogenic proteins 4 and 7 are expressed in the non-neural ectoderm at the border of the neural plate. So the sonic hedgehog protein signal repression at it signals when it's there, it represses the expression of PAX3 and PAX7, as well as MSX1 and MSX2. So what do you have? The sonic hedgehog protein is important in ventralization of the neural tube. So this protein that controls the ventralization is there to ensure that you now have the ventral portion of the spinal cord such that you have the basal plate differentiating there, and then you also have a floor plate at the midline. So the sonic hedgehog protein controls all that. This ventral region acquires the capacity to form the floor plate, like I said, and also the floor plate, which from where you have motor neurons differentiating. Sonic hedgehog protein is important as far as ventralization of the central of the spinal cord is concerned. But the bone morphogenic protein and 4 and 7 is the expression of bone morphogenic protein. Expression of bone morphogenic protein 4 and 7. It maintains and upregulates the PAX3 and PAX7 in the dorsal half of the neural tube, and you now have dorsalization there. So where you have sensory neurons in the alar plate, you now have sensory neurons formed. So you have genes that are required for formation of neural crest cells in the top of the neural folds, but their roles and those of the MSX genes in differentiation of sensory neurons and interneurons is not clear. However, their expression throughout the neural tube at early stages is essential for the formation of ventral cell types. You must know that even if their expression is excluded from ventral regions by the sonic hedgehog protein, at least you at these stages you have the ventral cell types that have competence to respond appropriately to the sonic hedgehog proteins, and then you also have other ventralizing signals they respond to. Then you have the PAX gene. The PAX6 is expressed throughout the elevating neural folds except in the midline, and this pattern is maintained. Now I need to talk about the applied embryology of development of the spinal cord. That's the clinical correlates. Very important, I need to talk about the neural tube defects. Most defects of the spinal cord result from abnormal closure of the neural tubes in the third and fourth weeks of development. You have the resulting abnormalities, which we call the neural tube defects, and this may involve the meninges, the vertebrae, muscles, and skin. You have severe neural tube defects involving neural and non-neural structures, and it occurs in 1 in 1,000 births. But the incidence could be high, like 1 in 100 births, like in places such as North China, because of the teratogens they were exposed to there as a result of the nuclear warfare. So the first neural tube defect I need to talk about here is spinal bifida. It's a general term for neural tube defects affecting the spinal region. In spinal bifida, you have splitting of the vertebral arches, and it may or may not involve underlying neural tissue. You have two different types of spinal bifida. You have the spinal bifida occulta and the spinal bifida cystica. Spinal bifida occulta is a defect in the vertebral arches that is covered by skin and usually does not involve underlying neural tissue. So spinal bifida occulta occurs in the lumbosacral region, that's L4 to S1 region, between the fourth lumbar vertebrae and the first sacral segment, and is usually marked by a patch of hair overlying the affected region. The defect is due to lack of fusion of the vertebral arches, and it affects about 10,000 of about 10% of people that you think are normal. They might not even know it. Then the second one I mentioned is spinal bifida cystica. It is a severe neural tube defect in which neural tissue, and you might also have meninges protruding through a defect in the vertebral arches, and it also protrudes through the skin to form a cyst-like sac. Most lie in the lumbosacral region, and you have neurological deficits, but they are usually not associated with mental retardation. In some cases, you can have a fluid-filled cavity with meninges protruding through the defect, and when you have that, we say the spinal bifida with meningeal. In some other cases, you have neural tissue, which it's in the sac, and then you have, we say it's not just the meninges protruding in the sac, you also have neural tissue, so we say it's spinal bifida with meningomyelocele. Occasionally you could have the neural folds not elevating at all, but they just remain as a flattened mass of neural tissue. We call that spinal bifida with myeloschisis and or rachis. Hydrocephaly develops in most cases of spinal bifida cystica because the spinal cord is tethered to the vertebral column. So as the vertebral column lengthens, what happens? The tethering pulls the cerebellum into the foramen magnum, and this interferes with the flow of cerebrospinal fluid into the spinal meninges, the subarachnoid space surrounding the spinal cord, and then you have the cerebrospinal fluid accumulating in the cranial meninges, and then you have hydrocephaly. Spinal bifida cystica can be diagnosed prenatally by ultrasound and by determination of alpha fetal protein levels in the maternal serum and amniotic fluid. So you can do it either radiologically by ultrasound or in the lab by sampling the maternal serum or amniotic fluid for the alpha fetal protein concentration. So that will tell you if the individual has spinal bifida cystica or not. The vertebral can be visualized by 12 weeks of gestation. You can do a radiological examination and see the vertebra, and defects in closure of the vertebral arches can be detected. You have treatment for spinal bifida. It could be by surgery. You could do the surgery in utero at about 28 weeks of gestation. So the baby is exposed by cesarean section, the defect is repaired and the infant is placed back in the uterus. And when this is done, you find out that when the baby is born, it improves the condition, so it might reduce the incidence of hydrocephalus and the other symptoms that the baby is supposed to feel. So what are the causes of spinal bifida? You could have hyperthermia, valproic acid, and maternal exposure to valproic acid, and then hypervitaminosis A, when the mother takes in excess of vitamin A. All this could produce neural tube defects. So you could also have a number of other teratogens. The origin of most neural tube defects is multifactorial, and the likelihood of having a child with a neural tube defect increases once one already has an affected offspring. You find out that it's been we are happier now because with the use of consumption of folic acid. When the pregnant woman takes in folic acid, it reduces the incidence of neural tube defects by as much as 70%. That's why a pregnant woman is advised to always take that. So if the 400 microgram of folic acid is taken daily beginning 2 months before conception, and you continue throughout the gestation period, it will reduce the likelihood of developing neural tube defects. So such a woman most unlikely will have a baby with neural tube defects. Thank you so much for listening. Don't forget to subscribe to this channel. Goodbye for now.